Welcome to Biology Made Easy. Today we are still solving genetic questions and for O-level and secondary school grade 10 to 12. Today we have questions on codominance or partial dominance and test cross. Welcome. In our question 1, red flowering snapdragons, they are plants, are homozygous for allele R1. White flowering snapdragons are homozygous for a different allele R2. Heterozygous plants R1, R2 bear pink flowers. What phenotypes should appear among the F1 offspring of crosses listed? A. R1, R1 cross R2, R2. B. R1, R2 cross R1, R2. This is how we analyze and solve the problem. Red flowering snapdragons are homozygous for allele. As soon as you say red flowering, you see there's going to be something red, and homozygous means this big R1, big R1. White flowering, white is here, and they are homozygous for a different allele. And this is it. So we have here white, and this is the allele. That's homozygous. Then heterozygous plants, that's R1, R2, bear pink flowers. So heterozygous, we write our heterozygous. Plants bear this genotype, then we put the genotype there. And we are being told that they bear pink flowers, so that is the pink. So this is how we are analyzing the text. Well, as soon as this red, white bears a heterozygous pink, you know codominance is in it. And of course, you should even see its codominance right from the beginning, because codominance, capital letters are used to represent the gene, and capital letters are also used to represent the allele. So this is codominance, right? And we are being told that A, we should cross R1, R1, crossing R2, R2. This is the cross. This is the genotypes have been supplied. These are the gametes. This one is all R1, R1 gametes that will be produced here. Here is all R2, R2. So the cross will give us R1, R2 genotypes, and we've been told the R1, R2, they are pink, all right? R1, R2 are pink. So this will give us this genotype and phenotype. So that is what we have, all pink. Now this cross, we have R1, R2 crossing R1, R2. The gametes that will be produced will be R1, R2, R1, R2. These are all the possible gametes. We cross the gametes using a Punnett square. These are the male gametes. These are the female gametes. These are the offsprings that will be produced. These offsprings, we write the F1 genotype. We have genotype R1, R1 is one of them. We have genotype R1, R2, and we have two of them, this and this. And we have genotype R2, R2, and we have 1. So we have genotypes 1:2:1, and we have phenotypes. This is red from up here, this is pink from up here, and this is white from up here. So these are the phenotypes. Good. For our second question, we have a dominant allele W confers black fur on guinea pigs. A guinea pig that is homozygous recessive, small w, has white fur. Fred would like to know whether his pet black-furred guinea pig is homozygous dominant or heterozygous. How might he determine his pet's genotype? As I always say, when you have a question, you break down the questions with genetic symbols. So a dominant allele W, we have our W written, confers black fur. So we come to see that W is black fur. A guinea pig that is homozygous recessive, this is WW, we write it down quickly, has white fur. So we see that WW is white fur. Fred would like to know whether his pet black-furred, so we write a black-furred here, guinea pig is homozygous, dominant, or heterozygous. So this is it. Fred wants to know this. How might he determine his pet's genotype? And if you know this is a test cross, he can determine the pet's genotype with a test cross where he crosses the phenotype with a homozygous recessive white. That's a test cross. So this is how we go about it. If the black fed is homozygous, that is, the parent is black, we have to cross it with a homozygous recessive white. So the parent phenotype is black, we are crossing it with white. Parent genotypes big W, crossing with small w. Meiosis takes place, gametes will be formed, and all the gametes that will be formed will be big W. All the gametes that will be formed with small w. So if we have a cross, mating, every cross will give us big W, small w. Every cross will give us big W, small w. And big W, small w will all give us black fed. So if Fred's pet is a homozygous dominant, big W, if we cross it with the homozygous recessive, all the offspring will be black fed. But if the black fed is heterozygous, the black fed is crossed with a white fed, that's the black, which is a heterozygous, crossed with a white fed, that's the parent, this will be small w. The gametes that will be produced are big W, small w. When we cross random fertilization, we can have big W crossing with this small w. We have big W, small w. This one can also cross. We have big W, small w. This will cross here, small w. This one will also fertilize with this small w. So the offspring will be half black, half white. So Fred can know whether his pet black fed is homozygous dominant or heterozygous if he crosses it with a homozygous recessive. All the offspring will be black fed. If his pet is a heterozygous, if he crosses it with a white fed, that's a test cross, he will have all the offspring, half of them black, half of them white. So what he can do is a test cross, and a test cross can also be known as a back cross. What that means is that he only has to cross his pet whose genotype he doesn't know with one of the parent, and that is the homozygous recessive parent. And so it's like going to cross with one of the parents. So it can also be regarded as a back cross. Well, this brings us to the end of our lesson for today. If the lesson has been useful, please give us your likes and subscribe. Thank you very much. Goodbye.